Katelyn Wasilenko, and to begin my journey towards military medicine and serving my country in the Army as a trauma doctor, I sought to improve bone repair through investigating a form of stem cell treatment. I investigated exosomes from pediatric stem cells for their ability to expedite bone healing, and I found that indeed exosome treatment offers a novel therapy to promote bone repair, with my principal finding leading me to the title of my research that pediatric bone mesenchymal stem cell exosome treatment requires interaction with the bone tissue defect environment to improve bone repair. The reason I explored exosome therapy is because currently, following painful fractures, orthopedic injuries, and surgeries in which healing is hampered by old age and disease like osteoporosis, the common clinical protocols involve vascularized bone grafting. Such procedures require multiple surgeries, and still 10% of all fractures remain a non-union injury. My treatment does not pose these complications, so you may ask, what was my treatment? Exosomes are characterized as the smallest of vesicles, 30 to 200 nanometers, released by all cells into what is called a secretome. Exosomes are extracellular vesicles that are essentially the UPS truck that transports proteins, DNA, and lipids from the cells in which they derive, the UPS facility, to currently unelucidated target cells, the homes that uptake them. My treatment revolved around exosomes released by pediatric bone mesenchymal stem cells and their role in the bone healing cascade. The molecular mechanisms of bone formation include the deposition, calcification, and maturation of osteoblast cells that bring the bone to a healed state. I studied the maturation of these osteoblasts with my novel exosome treatment when assessing its efficacy in vitro. I used exosomes as a treatment because Hofer et al. In 2016 found that the direct injection of bone mesenchymal stem cell exosomes is more effective than both the application of the entire stem cell itself and its larger secreted vesicles, microvesicles. Studies have found that these stem cells increase bone formation in rat models, and Z et al. In 2019 concluded that bone mesenchymal stem cell-derived exosomes increased proliferation and decreased apoptosis in osteoblasts. However, this study cultured the osteoblasts and exosome treatment in osteogenic media, which allows the exosomes to interact with more than just the osteoblasts because osteogenic media simulates the bone defect environment. So, my study sought to isolate this role, because the biological mechanisms of exosomes remain unclear, and the isolated role of osteoblasts and BMSC exos have never been studied. Additionally, my study brought in the whole other novel aspect of using pediatric exosomes. It was Malone et al. In 2011 that concluded that pediatric bone heals significantly faster than their adult counterparts due to increased bone density. However, no study, to my knowledge, has investigated the effect of exosomes deriving from pediatric bone marrow for bone repair. Thus, the purpose of my research was first to assess the impact of Bmsc exosome treatment on osteoblasts without supplementary media, and then compare the response to that of the treatments in bone mouse defects. My hypothesis was that the exosomes be more efficacious at promoting bone formation than other treatments, both in the osteoblast because they were indeed interacting with the osteoblast, and then in vivo in the mouse defect. For an overview of my methods, stem cells were extracted from pediatric surgical waste bone. In order to create treatments, I then carried out a series of centrifugations to remove pelleted larger contents and isolate the vesicles from cultured stem cells. The whole stem cells, microvesicles, and exosome factions were stored as treatment groups. These 3 groups were then applied in vitro to isolate osteoblast interactions, and then in vivo with the whole defect environment present. To study the isolated interactions between exosomes and osteoblasts, I applied these exosomes as treatments to MC3T3E1 cells, an osteoblast model, and then determined their maturation through alizarin red staining. MC3T3E1 cells at passages 3 to 6 were used in exosome-depleted FBS medium to ensure that the final media was void of vesicles naturally present within complete culture media and that only exosomes and osteoblasts could be interacting. For my in vivo aspect, my mentor created 3mm calvarial defects in mice and applied the 3 treatment groups with 2 mice in each group. I then analyzed micro-CT images taken 4 weeks following the 3 treatment group applications, and I used ImageJ software following the guidelines of Kuhn and Gao et al. In 2019. I calculated bone growth relative to the original 3mm defect and set thresholding at a minimum of 18 and a maximum of 225. All image analyses were performed in image J, and a Welch's unpaired t-test and one-way ANOVA were performed using GraphPad Prism 8 for statistical analysis. Significance was of course set at a p-value of 0.05. What I first found was a success of my pre-osteoblast cell culture. Along with morphological elongation of cells, the MC3T3E1 cell area was significantly increased from days 3 to day 7, meaning my cells themselves and my osteoblast models were indeed viable. Now, moving along to the mineralization of my MC3T3E1 pre-osteoblasts and their progression towards bone formation, I determined so using alizarin red staining, which assesses calcium deposition in cells. 2 weeks after application of treatments, when assessing the maturation of osteoblasts through calcium deposition, the lack of deep red staining in experimental groups in standard media, when compared to that of osteogenic media you see on the left, means that the exosomes and other treatment groups did not expedite osteoblast maturation. What this means is that it is not indeed the exosome interaction with osteoblasts that is specifically expediting bone formation. Now, my in vitro results contrasted that of the microCT image analysis from the in vivo application in mouse defects. 4 weeks following application of treatment, the defects with the PBMSC exos were significantly more healed than the defects by microvesicles with a p-value of 0.0003. They healed the bone after 4 weeks at 35.24%. Meanwhile, the PBMSCs as a whole only healed the bone and covered the defect at a rate of 9.88%. The improved efficacy of exosomes to increase bone formation in vivo, coupled with the lack of success in vitro, suggests that the bone defect environment is necessary to allow for PBMSC exos to communicate with other biological factors to mediate their efficacy other than the exosomes. It was Gao et al. In 2018 that validated the notion and my findings that osteogenic induction media increases osteoblast mineralization relative to standard growth and exosome-depleted media. Thus, the implementation of standard growth exosome-depleted media in this study was used to evaluate the efficacy of PBMSC exos without supplementary factors. Therefore, the findings of the study suggest that the communication of exosomes with other factors in the defect environment other than osteoblasts are critical to their efficacy. And I'm actually currently working on this. I want to know what other factors are indeed these exosomes are interacting with to promote bone repair, and I've learned R coding to perform differential gene expression analysis between RNA transcripts of exosome-treated groups to elucidate genetic changes and understand their interactions with other factors of bone healing, such as extracellular matrix deposition and angiogenesis. And I additionally am working to understand the difference specifically between adult and pediatric exosome treatment to see how, now that I found that the pediatric exosomes indeed do promote bone repair, to see how they match up to their adult counterparts. The reason that I'm doing this, and the whole reason for my project and isolate, is to isolate and store banks of exosomes in hospitals that can be injected as treatment for bone repair after orthopedic procedures, accidents, and with further hampered by osteoporosis. Additionally, the pediatric nature of my research has the aim of improving the gingivarioplasty procedure that fixes infants with cleft palates and typically leaves a gap in the skull following the procedure. With my research, implanting a biodegradable sponge scaffold injected with the pediatric exosomes in this gap will actually direct bone formation and expedite the formation and healing process to increase the efficacy of GPP procedures. To conclude my research, I employed novel methodology with pediatric bone mesenchymal stem cells. Their exosomes established 35.24% bone repair coverage and coverage of the defect after 4 weeks, as opposed to only 9.89 with the whole stem cells, which indicates that my exosomes can initiate osteogenesis and promote bone repair in vivo in a clinical setting. Since I determined it is not osteoblasts alone that these exosomes are interacting with to promote bone repair, further investigation, which I'm currently working on the secretome of PBMSCs, is necessary. Finally, I want to state that my exosome treatment is an efficacious means to facilitate bone regeneration with promising clinical outcomes. I would first like to thank my mentor and lab staff, my science research teachers for their unwavering support, and my peers in science research friends and family for supporting me along my journey of spending over 120 hours in lab conducting this research and then countless hours at home analyzing and compiling data. Thank you. Do you have any questions?